In this video, we are going to be talking about a few sequencing technology questions. There are some better examples of like looking at the data specifically within the warm-ups of the same day and the mid class of the Tuesday recording. Definitely look at those IGV images, make sure that you can interpret them in the way that we discussed in class. But here are some more ideas that you should be at least understanding after having watched this video before, like, say, we get to the test. And they're going to just help you understand some of these limitations that we're going to be investigating in our problem sets throughout this course. And indeed, if you end up ever working with sequencing data, they're going to come up. So the first question here is about the biggest issue with sequencing, one that is somewhat diminishing as we have developed longer and longer sequencing technologies that allow us to get information that's simply larger than the size of these challenging regions. And these challenging regions are often because of their repetitive nature. Here, I have described a region that is 250 base pairs and comprised of, even within itself, a repetitive sequence. So ATG, ATG, over and over and over. These regions occur for a couple of different reasons. There's some issues that happen if a chiasmata forms inside of them and you get this unequal crossing over and then they expand and contract. There's also this interesting phenomenon where the polymerase itself, while duplicating these regions, actually slips. It's like they kind of just ratchet up and down one unit of the repeat. I've actually taken advantage of this within my own research. This was my first ever graduate project, was investigating trinucleotide repeats in order to develop a fingerprint, a genetic fingerprint for different individual trees. In fact, I could determine pedigrees and identify crop like relative species instead of the pecan species I was working with. They're cool little places within the genome, but they also pose a problem. And one of those problems is that if your sequencing read is smaller than the size of the repeat region, especially because, well, they're essentially the same thing and they're all over our genomes, then any read that lands within it is useless to you. You don't know where in the genome it came from. It doesn't give you any extra information. It's just a waste. And so here, if you're really trying to identify what of these three different types of sequencing technology are the best, I recommend flipping this question on its head. The question may be easier to think about, like, which of these is most likely to touch unique information? Because unique information is the useful one, right? That's where we can actually map the read to the reference genome and figure out where it belongs and thus learn more about the genome. And so if you think about it in those terms, the one that's obviously always going to touch something unique is this much longer sequence. The 1,000 base pair sequence is always going to touch something unique. And so it's a great application for a region that contains a lot of repetitive or low complexity sequence. That's why that's been a game changer for our field, is that now that we have these expanded sequencing links, we're able to span these regions. The next best one will be these 75 base pair, then 200 base pair, and then another 75 base pair. This is the definition of a paired end read, where we get a sequence on one side of a fragment, a region of unknown DNA, and sequence on the other side as well. Now, for the record, in real life, if we already have access to 150 single end read, we probably have access to 150 paired end reads, but for the sake of making these somewhat a little bit more challenging, I made it 75 and 75. So these add up to this total same amount of sequence information, but this paired end simply spreads it out further. And it turns out, because it's then spreads it out larger than the repeat size, it's very, very rarely, unless the two repeats are very close to each other, it's rarely going to land both sides in a repeated region, whereas the 150 base pairs has a decent chance of landing in a base pair region. So by far, the 1,000 base pair is the best, somewhat better than the single paired end read, at least in the vast majority of scenarios. the paired end will be superior to the single end here. And it's because which one touches unique information. Next up, we have a coverage calculation. I'm going to pause the video really fast. And I'm back to calculate coverage. It's actually a very simple calculation. It looks complicated. It does have big numbers, but the actual calculation is pretty simple in concept. You take the total amount of sequencing information that you're able to generate, and put all of this, by the way, in the same unit. So anything base pair should just be in base pair. You have to fix like my megas and regular base pair and gigas and all that. But you get the total amount of sequencing information and divide it by how much you're spreading that out. So the genomic space. So it's the number of observations divided by the amount of things you need to observe. That gives you the average coverage, which is the average number of observations per unit of stuff you're observing. So in this situation, reading through it, we see 2 million paired end fragments. So that means each fragment will have one end and then the other end also sequenced. Each end is 50 base pairs. So that means 100 base pairs in total per fragment. So 200 times 100 should be my numerator. My denominator needs to be the genomic space. In this case, it's giga. So if you put everything in the same unit, so 2 million times 100 would be 2 times 10 to the 6th mega millions, the way to remember that. And then times 100. And then that would be divided by, well, if mega millions is 6, then gigas one level above that in our metric scale. So that would be 10 to the 9th. So 0.7 times 10 to the 9th. If I'd said terabase, then it would be 10 to the 12th, but I don't think there is anything with a terabase size genome yet that we know of. Terabase is only ever going to come up in some sort of like, compilation of data situation. But a lot of organisms have gigabase sized genomes, including us. Next up, we've got some information from one of my mutagenesis lines. And basically what we're looking at is read depth that has been transformed by an algorithm to determine the total number of copies of that region of the genome. Now, what this is actually based on is an analysis of that read depth estimated across the entire genome, which is currently known to be diploid. However, that does assume that there hasn't been a mutation so severe that it caused a full genome polyploidy. Now, for the record, I don't believe it's caused a full genome polyploidy, but I do believe a partial polyploidy has occurred, which is really cool. So here is the actual image we're looking at. We can see here median copy number on the left is hovering around 3 for these, and it's actually pretty consistent. We see a tiny little dip here where there's probably some average. So the way that this was generated was by looking at a 10 KB window, so a 10 kilo base, so 10,000 base pair window of measurements. And so this is kind of average, and that's why there's this little bit of intermittent. So this is just because of the calculation. As soon as we get to the other side of this mutation event and we get away from it far enough that that 10,000 base pair window is no longer estimating it, we're straight into diploid, which is what we expect for this species. And so what we're seeing here is a triploidy event. It's not a full triploidy, not a full trisomy, but it is a partial trisomy for this arm of the chromosome. Indeed, if we go to this link, we can actually see that there is a lot of stuff going on. Well, this one's not a lot, but this has a ton of stuff on the single chromosome, where we have large regions of that chromosome that have become trisomy. And a big question now is, well, where is it? And there are ways to do this. What we can do is, well, if I zoom in on here, what I can do is we can go into tools like that IGV software I was looking at, I was showing off in class, and we can look across this span of reads. And what we'll find is that there are some reads near the edges of this that reveal where this is coming from. They'll have like one side of the read will be aligning to this region, which there's just too many copies of. And then the other side will probably be attached to like a completely different chromosome. So what's probably happened is that there was a break in the DNA. It got repaired, but it ended up repairing it with an additional copy on accident. And then that copy got integrated into a different break site. All sorts of crazy things are happening, but somehow it just seems to have an additional copy of this section of the chromosome, which is really cool. Like, this is just cutting edge stuff. I did get scooped though. There was a popular paper that had just revealed the same information. So we haven't finished publishing it, but cool results, right? So we're getting these partial trisomies, and we're only now starting to uncover, well, what does that do? Plants are really robust to polyploidy events, like way more robust than animal systems are, just because of how they kind of balance the gene load, like how much a gene gets expressed. But this does affect them. And so, we would expect all sorts of behaviors. Like if you imagine there's a gene here that, let's say, promotes growth. Well, all of a sudden we have 3 copies of it. And so it may be produced at 150% of what it's normally produced at, which is fun. Like, that's pretty cool. But it's a complicated situation. So that's where we're at. Okay. So the question is, if the individual typically, so like for most of its genome where it's diploid, has 40 reads per locus, well, if you break that down, the read is coming because of sampling of the genome. And if there's 2 copies of most of the genome, then that would mean that on average, it's about 20 reads per copy per locus. And so if for this particular region we have 3 copies, then that would be 60. So the answer would be about 60. And that's correct. If you go and look at the read depth across that region, it is about 60. I can't remember if I included the actual numbers in my little summary information here. No. I was just wondering, I should go do that. That'd be a good thing to add. I started to run out of, I kept running out of memory. It's the issue. This is a pretty big data set. And so I had to do a lot of optimization. So I might have removed that value. So in the question above, what was best happening across the mutated region? Well, there was no deletion here. It's not a deletion across this region. A deletion where you would be left with a single copy would appear as like a bar down here. However, we're above, and so it's certainly a duplication at some point. I don't know what kind of duplication. I don't know where that duplicate copy got inserted. For all I know, that duplicate copy is just hanging out in the nucleus as like kind of a new temporary chromosome. Although I do know that this phenomena has lasted 1 generation of meiosis. Now, will it survive another generation of meiosis? I don't know. So that's some of these follow-up experiments, is finding the grandchildren of these sequenced individuals and seeing, do some of them still have these mutations? And there's all sorts of issues with like, well, no, the cells that had these mutations didn't manage to reproduce and stuff like that. But we'll find out. It's ongoing research. So definitely a duplication. And it says one duplication added 1 new copy or 2 new copies. Well, 2 new copies would end up adding 4. So that's definitely 1 new copy, becoming a partial trisomy. Okay, that's the end of the video. I hope that helped. Please reach out if you have any questions.